This lecture, part 1 of 4, will cover the structure and functions of cells. The topics of cell theory, the plasma membrane of cells, and diffusion and osmosis will be discussed. The human body is composed of trillions of cells. To gain an understanding of what a cell is, the first thing that needs to be examined is cell theory. There are 4 basic concepts to cell theory. First, cells are the building blocks of plants and animals. Cells are what these organisms are composed of. Second, cells are the smallest functioning units of life. Third, cells are produced through the division of pre-existing cells. And fourth, each cell maintains homeostasis, the state of having a stable internal environment. Cytology refers to the study of the structure and function of cells. It combines knowledge from several areas, including microscopic study, chemistry, and physics. Cytology starts with looking at the typical cell and its components. However, in reality, cells in the body are very diverse. Each cell type is specifically geared to perform specific functions. The diversity of cells found in the human body is demonstrated here in Figure 3-1. Cells come in many different shapes and sizes and have many different functions, from providing support to conducting electrical impulses to taking part in conception. The first component of the cell that will be examined is the plasma membrane, also known as the cell membrane. The cell membrane separates the cell from its environment and has 4 key functions. First, the cell membrane provides that physical isolation of the cell's components from the extracellular fluid that the cell resides in. Second, the cell membrane regulates the exchange of ions, nutrients, and wastes between the cell and its environment. Third, the cell membrane is sensitive to its environment. Any changes in the surrounding extracellular fluid will be noticed by the cell membrane first. And fourth, the cell membrane provides structural support for the cell. The cell membrane contains lipids, proteins, and carbohydrates. The cell membrane is composed of a phospholipid bilayer. Recall that phospholipids have a non-lipid head and fatty acid tails. The heads are water-loving or hydrophilic, and the tails are water-fearing or hydrophobic. As a result, phospholipids arrange themselves in a phospholipid bilayer with the heads facing out and the tails facing inward. That can be seen here, the phospholipid bilayer. We can see the polar heads and then the nonpolar tails facing inwards in that cell membrane. And that composes what is called that phospholipid bilayer. Substances such as oxygen, carbon dioxide, and other lipid-soluble molecules are able to easily diffuse across the membrane. Ions and water-soluble compounds cannot. Cholesterol, another lipid, can also be seen throughout the cell membrane in the regions of the fatty acid tails of the phospholipids. The cell membrane also has several membrane proteins, shown in blue in Figure 3-3. The membrane proteins include receptors, channels, carrier proteins, enzymes, and identifier proteins. The membrane proteins that extend across the entire plasma membrane are known as transmembrane proteins. The location and composition of the membrane proteins present can change over time based on the needs of the cell. It is important to note that the cell membrane is dynamic. It does not stay the same all the time based on its needs. It can go ahead and change its composition. Membrane carbohydrates, shown in purple in the diagram, are parts of more complex compounds known as glycoproteins and glycolipids. They serve the role of receptors. They also provide recognition for the immune system and can act as adhesive and lubricants. So here we go ahead and see some of those carbohydrate chains throughout the cell membrane. Permeability of the cell membrane determines what substances can cross the membrane. The term impermeable refers to when nothing can cross. The term freely permeable refers to when anything can cross. And the term selectively permeable refers to when some things can cross, but others cannot. The movement of substances across the membrane can be an active or a passive process. Active movement requires energy. For example, when moving a substance from an area of low concentration to an area of higher concentration. Passive movement does not require energy. For example, when a substance is moved from an area of high concentration to a lower area of concentration, an area of lower concentration. Molecules are continually on the move, and over time they will uniformly distribute. Diffusion is the movement of molecules from an area of high concentration to an area of low concentration. Again, diffusion is defined as the movement of molecules from an area of that high concentration to an area of lower concentration. The process of diffusion is passive, that is, no energy is required for this to occur. Molecules naturally will want to move down their concentration gradient from areas of higher concentration to areas of lower concentration. When diffusion across the cell membrane is considered, lipid-soluble molecules can easily directly diffuse across the phospholipid bilayer, but non-lipid soluble molecules cannot. This makes the cell membrane selectively permeable. Instead, non-lipid soluble molecules must diffuse through other means, such as channel proteins that are found in the cell membrane. The diagram below represents diffusion. A color of cube sugar is dropped in the beaker of water. As it dissolves, the concentration of sugar near the bottom of the beaker increases. The sugar will diffuse from this region of high concentration. So here we have high concentration of that sugar to an area of low concentration of sugar at the top of the beaker. So diffusion will go ahead and occur down the concentration gradient from high concentration to low concentration. Over time, a uniform distribution will be achieved and a concentration gradient will no longer exist, as shown in this beaker here. Here that sugar has diffused throughout the beaker equally. Now there is no concentration gradient as the concentration of sugar is the same throughout, so there will be no net movement, there will be no diffusion occurring. Both the size and lipid solubility of a substance are variables that will determine how substances can cross the cell membrane. Lipid soluble molecules are free to diffuse across the membrane, as seen here. So something like a steroid, a lipid, which is going to be soluble across that phospholipid bilayer, can easily go into and back out of the cell. Large molecules, such as the one seen here, will not be able to pass through the cell membrane and will have to rely on the aid of carrier mechanisms to get them across the membrane. Finally, small water-soluble molecules take the route of diffusion in and out of channel proteins that are found in the cell membrane. Let's take a look at osmosis. Osmosis is the diffusion of water across a selectively permeable membrane, meaning that water can diffuse across the membrane, but solutes cannot. Water will move from the low solute concentration to a high solute concentration. In other words, water will move to areas with more particles. You can also think of this as water moving from a place where water is highly concentrated to a place where water is in a lower concentration. This corresponds to the area that has more solute particles. If we have more solute particles compared in one region compared to another, that area will have a lower concentration of water. As a result, water will move from the area with fewer particles, where its concentration is greater, to the area where there are more particles, where that concentration of that water is smaller. This is easiest to remember in that water will flow towards the area with more particles or more solutes. The following diagram illustrates the concepts of osmosis. Here, two solutions are separated by a selectively permeable membrane, seen here. We have solution A and solution B. Since side A has a lower solute concentration compared to side B, water can freely flow across that selectively permeable membrane and will move towards side B where the concentration of solutes is greater. Water will go to the area that houses more particles, more solutes. Let's go ahead and check if this reasoning is correct. Indeed, water will move across the selectively permeable membrane to side B. This concept is important because the solute concentration inside and outside of a living cell, along with the movement of water, can affect the shape of the cell. So again, let's review that one more time. So here on side A, we have fewer solute particles compared to side B, where there are more. As a result, the concentration of water will be higher on this side compared to the concentration of water on side B. Water will want to flow towards the molecules from the area of high water concentration to the area of low water concentration, and because the selectively permeable membrane does not allow the solutes or particles to move, we'll have the water flowing towards the area of more of those particles, more of those solutes. So water will go ahead and flow from A to side B. And again, that was demonstrated here. Water has, the volume on side A has decreased, the volume on side B has increased because that water went ahead, traveled through that selectively permeable membrane towards the region that had more of those particles. In what is known as an isotonic solution, the concentration of solutes on either side of the membrane will be equal. As a result, there will be no net movement of water into or out of the cell. In a hypotonic solution, the concentration of solutes outside the cell is lower than inside the cell. As a result, water will move into the cell. In a hypertonic solution, the concentration of solutes is greater outside the cell than inside the cell. As a result, water moves out of the cell. Let's take a look at this visually to gain a better understanding. This figure demonstrates what will happen to the shape of a red blood cell when it is exposed to solutions having different tonicity. In an isotonic solution, seen here, the solute concentration is equal both inside and outside of the cell. As a result, water will be moving into the cell and out of the cell at the same rate, and no net movement will occur. The cell retains its normal shape. In a hypotonic solution, there are fewer solutes outside the cell compared to inside the cell. So here we see some of those solute particles that are inside the cell, whereas outside of the cell, in a hypotonic solution, there are a relatively lower amount, in this case no particles present. As a result, water will move towards the area of higher concentration of solutes, in this case into the cell, causing the cell to swell and possibly burst or lyse. This is what happens in a hypotonic solution. Finally, when we consider a hypertonic solution, hypertonic, there are more solutes outside the cell compared to inside the cell. So here we can see the cell has a few solute particles, but the solution, the hypertonic solution it sits in, has many, many, many particles of that solute dispersed throughout it. Here, water again will move towards the higher concentration of those solutes. Water follows those particles, this time resulting in water leaving the cell. So we can see water is going to flow from inside the cell outside towards that area of high solute concentration. The cell then shrivels with the loss of water and crenation occurs. So to review, here's an example of an isotonic solution, a hypotonic solution, and a hypertonic solution. I always remember that the cells swell In a hypotonic solution, the O in hypotonic indicates that in the hypo, that swelling of that cell. Something to help you remember. Let's go ahead and review with a checkpoint. Can you remember the 4 basic concepts of cell theory? Go ahead and pause the lecture while you formulate your response. The 4 concepts of cell theory are: cells are the building blocks of plants and animals. They're the smallest functioning units of life. Cells are produced through the division of pre-existing cells, and each individual cell will maintain homeostasis. Let's try one more. What is meant by selectively permeable when referring to a plasma membrane? Selectively permeable refers to when some substances are able to diffuse across a cell or plasma membrane while others are not.